Okay, we're in the home stretch. This has been a really fascinating series of panels, and we're going to do a wrap-up panel now, followed by some closing comments from our director and from my Vicky Whittemore, who's also helped us organize this conference. So it's a real pleasure for me to introduce Dr. Susan Masino, who is the Vernon D. Russo Professor of Applied Science at Trinity College. Her research focuses on promoting and restoring brain health with a particular interest in adenosine and on the relationship between metabolism, brain activity, and behavior. Dr. Masino.
Thank you very much, Dr. Hartman. So I'm delighted to chair this wrap-up panel of this really historic and important conference today. I'll just briefly introduce the participants, who have all been participating today, but in case you missed it, Dr. Mackenzie Cervenka, Associate Professor of Neurology at Johns Hopkins University School of Medicine, Medical Director of the Johns Hopkins Adult Epilepsy Diet Center and the Adult Epilepsy Monitoring Unit. Dr. Juan Pascual, inaugural holder of the Once Upon a Time Foundation Professorship in Pediatric Neurologic Diseases and Professorship in Neurology. Dr. Jong, Professor of Neurosciences and Pediatrics at University of California, San Diego, and the Division Chief of Pediatric Neurology at the Rady Children's Hospital in San Diego. Dr. Elizabeth Thiele, neurologist and epileptologist at Massachusetts General Hospital, Director of the Pediatric Epilepsy Service, and Professor in Neurology at Harvard Medical School. And Dr. Gary Yellen, Professor of Neurobiology at Harvard Medical School in Boston. And he is a basic scientist and has been looking at the interplay between cellular brain metabolism and brain excitability using basic science research tools, and in fact, in some cases, developing those tools. So we have a short amount of time and a fascinating group of kind of clinical, translational, and basic scientists on the panel. So what I'd like to do is to offer some questions to kind of focus our thinking about maybe the near term to 5-year goals and maybe a longer term 20-year goal. And some of the things I'm going to integrate into this panel are sort of follow-up things on previous panels. So related to the last panel, I really appreciated the recognition of the presence of outliers and the importance of observational work. So related to where we are clinically, I'll just kind of give an opening question to the panel. Should the community work together to develop standards to improve the rigor of clinical work and observations, so deal with underpowered findings, look at more consistent groups of patients or sort them as possible, explicitly recognize the opportunities afforded by outliers, and if so, are there human studies that you think really need to be done? And are there basic or clinical tools that are needed right now or could be improved? And maybe I'll just start with Dr. Cervenka.
Yes, thank you, Dr. Masino. So I think that is a terrific question, and the simple answer I would say is yes. Yes, I think we need all of those things. I think there absolutely are areas where this can be approached clinically in large-scale clinical trials. The one of particular interest, and it's interesting because there's actually a question that just came up in the Q&A as well that pertains to this, is in the condition of status epilepticus, so super refractory status epilepticus. And what we know from the pediatric literature is that there is a condition called FIRES, and a comparable condition called NORS, new onset refractory status epilepticus, which, discussions are about whether or not those are actually a continuum of the same condition as well, and whether this is a population that benefit particularly from ketogenic diet therapies, perhaps with regards to the anti-inflammatory properties of the ketogenic diet therapies, both from the mechanism of what we've heard about with glucose and also with ketone bodies being protective in addition to that. So I think this is a potential opportunity to look at larger scale clinical trials and be able to start to delve into some of those those basic mechanisms as well, because it is such a specific population. And where we know that there are groups that are very responsive in some of these observational studies, some of which that have been retrospective, the response rate is sometimes as high as 90%, which when you compare that to, as the person pointed out in the Q&A, mortality rates of 30 and 40%, a 90% response rate, meaning resolution of status epilepticus, is pretty dramatic.
Thank you. Dr. Pascual.
Yeah, thank you so much. I think there are three aspects that are being implemented in other areas of scientific inquiry, and I think that they should be of some value if they come to the clinical trial arena. Number one, many of the journals where you publish scientific data will already append or offer or attached critique by an expert, where you look at the difficulties and the caveats and the limitations. Clinical journals not always do that. Number two, also some of the journals already published the peer reviews with the article, just to see what exactly went through the mind of the people who really thought about the issues really hard and carefully. And number three, many of these clinical trials do not publish the full data set for people to come in and do their own analysis. So if I'm interested in a particular type of responsiveness to an intervention in a particular kind of epilepsy, and I want to sample or look at the 10% people who have a great response, even though the trial might have been negative, right now I have no easy way to access that data. I like to look at that and be able to be very, it should be seamless to everybody, and the format should be workable in some way. We just don't have that yet.
Thank you. Dr. Rowe.
So I would follow up on Dr. Pascual's comment that I think it would be worthwhile strongly considering looking at the so-called super responders to the ketogenic diet and maybe focusing clinical studies to ascertain what is unique about them that makes them so responsive. In contrast, it would also be worthwhile to look at those who fail a response and maybe even have their epilepsies worsened by ketogenic diet. And I know that there aren't too many patients like that, and there are certain contraindications, obviously, with certain fatty acid oxidation disorders, et cetera. But there are intriguing reports out there of certain mutations. For example, SLC 13A5, which is a citrate transporter. That it's a rare condition, but it turns out, at least from reports, that perhaps the ketogenic diet may actually worsen the epileptic condition. So I think looking on both ends would be highly instructive in terms of giving us some clues as to what determines efficacy or the reverse.
Thank you. Dr. Thiele.
I think I would agree with what everyone else has said. I think there's a real need for better understanding the role of dietary therapy in epilepsy. My career has been rare epilepsies. So I love the idea of basket studies, to try and learn with this smaller group of placebo people to try and learn kind of, is it particularly effective in which populations? And then that may also allow us the opportunity to look at super responders. Like, why would kids with Angelman syndrome, why would diet be more effective for them and maybe not as effective in other disorders? So I like that. I also agree with Mackenzie in the comment that super refractory status epilepticus, all of us that do diet, we all have anecdotal stories of it working in that setting. But here's where it becomes a little bit more difficult, is doing a trial with diet is not as easy as doing a pharmaceutical trial. Because the calculation of the diet, the availability of the dietitians, very few of us have 24/7 dietitians. So I think the trial design in that setting would really have to take all of those different factors into account. But I agree. I think the diet can be wicked effective in super refractory status epilepticus. And so it would be good, since there's a huge unmet need for effective, safe, and well-tolerated treatments there.
Thank you. Dr. Yellen.
Well, as the other non-clinician in the group, I kind of an outsider to this particular question, but I think your question about outliers is pretty interesting. And it's clearly an example. I mean, if they're outliers, we're not going to get any reliable, statistically significant, clinical trial information out of those outliers. But this is a case where I think gifted clinicians are really at an advantage. You know, people who see the picture and who might look at an isolated fact, maybe you know, a citrate transporter mutation in one patient, and just say, gee, I wonder if that has something to do with it. And this is hypothesis generating. It's exactly the type of paradigm where we can move stuff from a clinical observation back to basic science, where we can test it much more systematically. But I think, you know, it's still, you know, looking at outliers is yes, you can do it in the context of a clinical trial, but I think it really points to, you know, maybe sometimes unrecognized genius among clinicians for seeing a pattern on the basis of small evidence.
So, Susan, if I could just add to that, I think one of the things I always tell medical students and residents when I talk to them or work with them is I think it's really important when you're taking care of patients to take a step back and try and figure out what the patients are trying to teach you. And it was really that observation and doing that what led us to be able to develop low glycemic index treatment, because we realized that it was glucose doing this, and these new kids would have rapid breakthrough seizures, and thinking maybe that was part of the mechanism. So I think that's important, and I think that we can all learn more about diet and help direct basic science by what observations we're making clinically about many things: who's really responding, who's not responding. You know, why would it be so effective in super refractory status, which is wicked difficult to treat, you know, et cetera.
Mm-hmm. Yeah, and just to tag on to the point that you just made is thinking about what promotes efficacy and then what also causes the reversibility as the other side of the mechanism. So I'm going to ask another question. It's kind of a team science question, and in the interest of time, maybe try and keep your responses a little bit short so that we can have time for another question. But lately I've been working on a team science project in Connecticut related to the Governor's Council on Climate Change. And we've been talking about using the principles of multi-solving, which was developed to kind of deal with climate problems. So with multi-solving, having a climate benefit is always a core benefit. But you're also looking for other near-term benefits. In the last panel, there was a discussion of, is there more cognitive benefits or mood benefits or ease of administration, you know, all these other kind of co-benefits that we often talk about. And one of the core principles of multi-solving is that you'd want to bring the whole system together to look at it, that no one perspective would dominate and conflicts of interest wouldn't dominate either. So if I thought about this from a team science approach, you know, we'd want to think about something that of course dealt with the primary clinical problem, but also at the same time consider these other benefits and pull together a team that has those qualities, that no perspective or turf dominates and conflicts of interest or any kind of rewards or something doesn't dominate. And I wanted to see what you thought about trying to really pull together a team that could operate under those principles. And one other principle is you're always trying to solve other problems that other parts of the system bring in. So the patient or the basic scientist or whatever. So anyway, maybe I'll go in reverse order on this one right now and start with Dr. Yellen.
Well, I'm not sure, Susan, because you ask such a big umbrella question. I think it's a great idea to basically have a lot of eyes on a problem, and you know, both a lot of perspectives and a lot of potential outputs. But I'm not sure exactly what you're suggesting in the context of this particular problem. So I'm going to just pass and let other people tackle it.
All right. I'll pass it on to Elizabeth. If she doesn't understand the question, also I'll rephrase it.
I think I understand the question, and I think that I've always been impressed that the ketogenic diet community, both the clinicians and basic scientists, have overall had a really nice relationship and work together, share ideas. My dear friend Vicky Whittemore has a great experience with this, with the TS kind of tuberous sclerosis alliance community of people working together to try and, you know, not have walls between us to improve our understanding of things. So I think that is happening, and I think keeping the channels of communication widely open and this go between the translation between, even if I don't have a biomarker for something, what am I seeing and what can that mean? So I think that the communication and the dialogue is a really, really important aspect.
Thank you. Dr. Rowe.
Well, so I highly support the concept of devising a quote unquote team science approach. Obviously, people define these differently and implement them differently. But if we take a more recent model that was pioneered by CURE, Citizens United for Research in Epilepsy in Chicago, where they launched a pilot project a number of years ago, trying to pursue a team science approach towards developing a novel therapeutic for infantile spasms. That white paper actually has just been accepted and will be published in, I believe, Epilepsy Open, if I'm not mistaken. And so this describes the kind of pulling together of basic translational scientists, clinical researchers in a team framework after a single goal of identifying a novel therapeutic that they could ultimately validate later in human studies. While the clinical study hasn't been done yet, there are some interesting lessons learned and approaches taken with that kind of team science approach. Now, this could be applied to clinical researchers, tackling maybe a fundamental problem in the clinical arena, or it could be, bench scientists who, come together and say, well, let's look at all aspects of sort of the mechanistic hypotheses, whether it's microbiome related, whether it's, amino acid, branch chain amino acid supplementation, et cetera. Maybe identifying a single model if they agree that could be dissected, even though every model has its imperfections. And then maybe bring those two together as some approach to get some new insights into what's happening with the diet and what the underlying mechanisms are. So, this is a different paradigm. I know the NIH has kind of pioneered many funding mechanisms and opportunities to try to bring people together from different level, areas of expertise. And some have worked well and some have not. And we don't have anything currently like that for the ketogenic diet or metabolism-based treatments. And I realize we're split fairly widely globally, but perhaps there is a novel way we could, approach some type of team science approach. So I support that concept very fully.
All right, Dr. Pascual.
Well, thank you. As much as I love all of my colleagues and I appreciate everything you're saying, I will dissent. And I would argue that in reality, if you look at the many things that have failed, and you do error analysis and wonder why so many things have really failed, the answer to that is not in what they're in how they're different. It's actually in what they all have in common. The assumptions, the hypothesis, a very common root understanding of the problems we're dealing with. And so group science has a tendency to unify concepts. You have a tendency to make everything homogeneous and agreeable by consensus. So if you look at how penicillin, X-rays, MRI, CRISPR were invented, they're not the product of team science, they're the product of some scientist, mostly forgotten somewhere, who's working on some silly invertebrate that nobody really care about. And then some breakthrough came along, and then a number of clever people follow through. So I agree with team science for a number of questions, of course, no question about it, but I also believe in the freedom of the individual to do sheer plain discovery.
Mm-hmm. Yeah, the power.
Yeah.
Oh, sorry. Go ahead, Jong.
Can I respond to that? No. So
Sure.
they're not meant to be mutually exclusive. Obviously, you could do parallel approaches. I think the best example in clinical medicine of a sort of team effort to advance science and therapeutic efficacy is the oncology trials. Where they, the COG or the Children's Oncology Group, for example. If you look back decades ago, childhood leukemia was a death sentence. But with the way they approach things systematically together, taking novel, therapeutics as they were being developed and applying them, in the homogeneous target populations, they've been able to turn the death sentence into a 90 to 95% survival, right? So that's not something we've done in epilepsy. And I think that there's some lessons that we can take from the oncology field that could help, not at the exclusion of, independent scientific discovery, which oftentimes, as you know, comes as a product of serendipity. But nevertheless, I think both should be allowed to flourish and foster.
1 minute remaining.
Okay, we just have 1 minute remaining. I want to see if Dr. Cervenka has anything to add, and I'm going to ask a final yes or no question. Dr. Cervenka.
I'll be very brief. I guess one consideration might be to expand this. I know this is with the focus on epilepsy, but as we're talking about ketogenic diet therapies, there may be other neurologic disorders, or there are other neurologic disorders where this is very relevant. And so when we're thinking of this team approach, maybe opening it to some of these other groups, because they might hear about some of the mechanisms that we're studying that could not be relevant to epilepsy, but may be relevant to other areas. Thank you.
Totally agree. Okay, we're on to the final lightning round, yes or no question. What are your thoughts on a poly pill, given the plethora of mechanisms? And I think there hasn't been a lot of approaches in terms of looking at synergies between these mechanisms. And some mechanisms can be mobilized by relatively non-toxic treatments. So ideas on the poly pill, I will call out your last name in alphabetical order. Cervenka.
Too soon.
Pascual.
Hopefully, yes. I mean, every other disease, chronic disease, is handled through a variety of interventions at the same time.
Ro.
I think it's going to become a combination of a big horse pill plus some form of diet, at least for the immediate future.
Agree. Thiele.
Agree too soon, but it'd be pretty wicked awesome.
Agree. Yellen.
Very complicated to optimize.
True. Big data space. Thank you so much to everyone. And I think that wraps up our panel, and we will be going to the closing comments.
It's wonderful. Thanks a million to all of our panelists throughout the entire day. It's been fantastic, and we're really appreciative of you putting your heads into all of these really challenging questions. So our next speaker is going to be the Deputy Director of NINDS, and it's a real pleasure for me to introduce her. Dr. Nina Schor is the Deputy Director of NINDS and a Senior Faculty Associate at the University of Rochester. During her fellowship, she began her studies of neuroblastoma aimed at understanding the neurobiology of this tumor and exploiting this understanding to design and test in preclinical models novel strategies for the therapy of chemo-resistant neuroblastoma. For the next 20 years, Dr. Schor rose through the academic and administrative ranks at the University of Pittsburgh, ultimately becoming the Carol Ann Cromer Professor of Pediatric Research, Chief of the Division of Child Neurology in the Department of Pediatrics, and Associate Dean for Medical Student Research at the Medical School. In 2006, Dr. Schor became the William H. Eilinger Chair of the Department of Pediatrics and Pediatrician-in-Chief of the Golisano Children's Hospital at the University of Rochester, post that she held until January 2018, when she became our Deputy Director at NINDS. Dr. Schor.
Thank you, Adam, and thanks to all of you for such a wonderful set of discussions. I'm going to pull up some slides that I hope will help us put together some of the presentations that you provided ahead of time with some of the comments that were made during the discussion. I really must thank you for being willing to tackle as a group what is one of the messiest problems that child neurologists face. What I would like to do is use one or two of the pre-recorded talks to illustrate some of the points that were made during the discussion. So you had a discussion of how work on basic mechanisms informs the development of treatments and prevention methods for epilepsy, and how rigor can be improved. And I want to use the microbiome to give you a sense of just how incredibly complex this is and what challenges face us, and why synergy, as occurs in a group that comes at this problem from different vantage points, is really the only way to tackle it. This is a diagram of what happens in epilepsy and what we do to try to thwart it. And in the midst of this is the microbiome, its effects on and the effects of diet, genetics, inflammation, drugs that we throw at epilepsy. Drugs influence the microbiome and are influenced by the microbiome. The microbiome influences brain dysfunction, perhaps SUDEP as well, but clearly the incidence of brain dysfunction and the systemic disruption that occurs because of it changes the microbiome. And it is in the midst of all of these effects, some of them endogenous and some of them exogenous, some of them intending to reverse the epileptic diathesis, and others of them unrelated but occurring in the setting of the epileptic diathesis. This bidirectionality of every single one of these factors makes making sense out of observations in the midst of a clinical study very difficult. And I've tried to summarize the role of the microbiome by saying you are what you eat what you are eats you, and what eats you influences what you are. And this circularity, this notion that you cannot make an intervention, whether you're doing it because of the problem you're tackling or for other reasons, you cannot make an intervention without modulating almost every other parameter at which you might be looking. It's almost like the Heisenberg uncertainty principle. And so separating out correlation from causality and cause from effect becomes very, very difficult, even in the setting of optimally rigorous studies. And we heard that in several of the discussions today. You discussed how information from patients can inform mechanism studies and how we can optimize translation to promote both basic and clinical studies. And I'm going to use biomarkers as an illustrator of the challenges of doing this. So again, here's a diagram. There are some people who are susceptible for whatever reason to epilepsy. Some of them go on to develop epilepsy. Sometimes the epilepsy progresses. Sometimes we throw drugs at it, or the natural history dictates that the epilepsy improves. Sometimes the drugs have side effects, and sometimes the drugs are simply not effective. In all of these cases, we could go on to develop biomarkers for each of these processes. We could say, how do we know that a patient has developed epilepsy? And maybe the EEG is the biomarker we use. Or how do we separate out the patients who improve from the patients who don't? And maybe we look at an electrographic marker or a behavioral marker or a biochemical marker to say that this population improved and this population didn't. And one could say that any one of those biomarkers is a prospective target for therapy. And that would make the world very simple. But the problem, as you've heard several times today, is that some of those biomarkers are not really biomarkers. They're surrogates. They're true, and not related to etiology. They may be epiphenomena. They may be effects rather than causes. And so aiming a therapy at something that is not really a biomarker means that you are aiming a therapy at something that should not be a target. And separating these out becomes very, very difficult in the setting of all of the observations we make in our patients. How do you know effect from etiology? And then finally, you discussed what questions can be answered by clinical trials and how you design these clinical trials. And the name of this conference is Metabolism-based Therapies, but I would challenge any of you to name something that occurs in a living, awake, behaving organism of any sort that is not metabolism based, or at least metabolism related. And this is my, I'll tell you that I was a biochemistry major, and I would shock many of my professors if they saw this diagram, but you can see the Krebs cycle somewhere in here in the blue, and the urea cycle and all of the feedback pathways. But in the midst of this complex diagram that is metabolism, we throw diet, development, aging, exercise, pregnancy, fever, inflammation, drug therapies, and histone methylation, which affects the transcriptome. And so there are many, many things for which in a clinical trial, it is very difficult, if not impossible, to control, that try though we might, we cannot get around the fact that they affect every measurement that we could possibly make. And so given that metabolism is life and life is metabolism, how do we decide if an alteration of the course of epilepsy is related to the perturbation that we make in metabolism, or is an epiphenomenon thereof, or is a consequence of intervention unrelated natural history of the disease? And in the setting of this, how do you pick a good control group? And you've already talked today about platform trials and about pragmatic paradigms that really get around this issue. What are you going to do? Are you going to take patients off of all their medications to create a true control group and make the study clean? You cannot. And so doing clinical trials becomes the challenge of coming up with innovative ways, not to get around it, but to make metabolism work with those things that we already know improve the patient's outlook. So what is going to make a difference for our patients? And I think that there are a few things that we can say make a difference for our patients. I think, and many of you have said this today, understanding the mechanisms of the role of metabolic interventions in changing susceptibility to epilepsy. We really should be looking at epileptogenesis and not at stopping seizures after the fact. And I know this is the holy grail in everything that we do. The role of metabolic interventions in changing the likelihood and severity of seizures in those who are susceptible to or who have epilepsy. The role of metabolic interventions in determination of effectiveness and toxicity of the drugs that we use or the devices that we use as therapeutic approaches to epilepsy. What makes some people respond and some people not respond? And the role of metabolic perturbations in resilience to seizures in individuals who are susceptible to epilepsy. How come some people have the same genetic substrate and the same environmental upbringing and don't develop epilepsy, while a sibling, for example, or a twin may develop epilepsy? And then from the standpoint of how we make this relevant and important for patients and families, how do we disseminate the information we gather on metabolic perturbations and epilepsy? How do we determine not only what works and what doesn't, and what's tractable and what's not in the context of family life of all of our patients, but how do we decide that a particular change that we induce is meaningful for that patient and family? How do we make sure that we treat the things that really are a problem for these children? Well, I think I'd be remiss in the setting of all of this if I didn't thank you again, really, for being our partners in helping us carry out our mission to use science and to use research to ease the burden of neurological disorders. Thank you so much.
Well, thanks a million, Nina. That was wonderful. So our final comments, our wrap-up comments here are going to be from one of my partners in crime, Dr. Vicky Whittemore. Dr. Whittemore is a program director at NINDS in the Synapses and Channels Cluster in the Division of Neuroscience. There she oversees a grant portfolio that includes basic translational and clinical studies on epilepsy. She also oversees a grant portfolio on myalgic encephalomyelitis and chronic fatigue syndrome and fatigue. Her additional interests are in research on global health issues, stigma, and comorbidities. Dr. Whittemore, bring us home.
Thank you, Adam, and I'll be as brief as I can here, but just to give you my sort of personal perspective and thoughts about the day. First, I'd like to thank the panelists for really thoughtful discussions and being open and honest about facing what the challenges are in doing research on metabolic therapies for the epilepsies, and the participants for lots of the amazing questions that have come in. Gives us a lot of food for thought. And it's my perspective that this is just the first conversation, that this workshop has really opened up an avenue for discussion about how to move forward in terms of how do we better understand mechanisms to better translate that understanding to clinical trials and therapies for individuals with epilepsy. So I approach my comments sort of from two perspectives. First, from a personal perspective, because I've known many individuals who have significantly benefited from dietary therapies that have really helped to control their epilepsy. And sure, it's not easy and it's challenging for them as well as their families. But when you see the amazing results that you can get, it makes it very much worthwhile. And I would love to see us learn, as has been discussed here, learn more from what is it about the people that respond, what is it that's changing in their metabolism that they respond and it stops the seizures. So the other perspective, the other perspective is from a scientific and personal perspective. So as Susan knows, I had a dog, a husky, who had epilepsy. He had a grand mal, or clonic, what do you call them now? A tonic-clonic seizure, generalized tonic-clonic seizure. And medicine just did not work well for him. So we put him on essentially what I would call a modified Atkins diet. And over about the course of the last 5 years of his life, he was seizure-free on that diet, except for the 2 times when my kids fed him pizza crust and he had seizures. But if there had been a research study, I would have gladly enrolled him in that study to see what's changing in his metabolism. How, if you do challenge him with carbohydrates, does it lead to a seizure? And I think that there are probably a lot of other people out there like me who would do that. From the scientific perspective, I would love to understand what's going on. And I think as there are so many causes of epilepsy, I think it's very likely that there's not one underlying mechanism through which the metabolic therapies for epilepsy is working. But I think if we all come together and really think carefully about how to move forward in this field to both gain information where we can from animal models that are closer to humans, like dogs, or from our subjects, our study participants, or our patients, we can really make progress in this field. And again, I'd like to thank all of you for your really thoughtful comments. It's been a really interesting and I think fruitful workshop. At last, I'll thank Adam for all the hard work he put into organizing this workshop and making sure everything was in place to make this really a terrific workshop. So with that, I'll turn it back over to you, Adam.
Well, thanks a million, Vicky. This has been really, it's one of those mind-blowing kinds of conferences. And so I think that we have a lot of work to do, right? So I first wanted to mention that the pre-conference videos are available on the website. And I posted that earlier in the chat. Please do look at them. And maybe now I'm almost wondering whether I should go back and look at them again after hearing the discussions today. We also, the videos from today also will be available probably in a couple of weeks on the NINDS website. We hope to keep those up for as long as possible. They need to be captioned, and so we need a little bit of time to get those prepared. I wanted to just extend a few thanks. The first is to our patients and families who really have served as the inspiration for all of the wonderful clinical work, translational work, and basic science work. This has really been, you're what really drives us. I wanted to thank our organizing committee for all of their hard work and for my pestering emails at all hours of the day. And I wanted to thank our panelists for all of their hard work today as well. And finally, thank our leadership, Drs. Koroshetz and Schor, for their support in allowing us to move this forward. So thank you all very much. As I mentioned at the beginning, the trains around here run on time, and so we have an on-time arrival. Thanks a million to everyone. And we look forward to hearing more about the work that's going on in this area. Thanks again. Have a great week.